Hello everyone, in this video we'll be discussing about chemiosmotic hypothesis. Wheresoever in nature ATP production will occur, you would be seeing a proton gradient being formed. And when this proton gradient is broken down, the energy released by this process is coupled with the formation of ATP. So this is explained by a hypothesis which is called as chemiosmotic hypothesis. So let's discuss this. We have discussed this hypothesis in respiration also, and now we are discussing here in photosynthesis. Now, and we'll be discussing what is the difference between the proton gradient in respiration and in photosynthesis. So what does this chemiosmotic hypothesis do? It explains the synthesis of ATP. It is linked to the development of proton gradient. Proton means H plus ions across a membrane. And when this gradient will break down, the energy release would be trapped in the form of ATP. Now let's discuss the differentiating features of this proton gradient between respiration and photosynthesis. In respiration, the proton gradient was between the two membranes, that is the intermembrane space of mitochondria, between outer membrane and the inner membrane. But in photosynthesis, the protons are accumulated in the lumen of thylakoid. Thylakoid are the disc-like structure present inside the stroma of chloroplast. Now let us see how this development of proton gradient in photosynthesis occur. We have by far come to know that in respiration these protons were being accumulated between the two membranes of mitochondria. But in photosynthesis the protons are being accumulated inside the lumen of thylakoid. And there are three important processes which lead to the formation or say accumulation of hydrogen ions inside the lumen of thylakoid. First of all, the generous water would be splitting. The two molecules of water would split into 4 hydrogen atoms, or say 4 protons plus 4 electrons and one complete molecule of oxygen. So splitting of water takes place in the lumen of thylakoid. Therefore, these protons are accumulated inside the lumen of thylakoid. Now the second thing is, during electron transport, as electrons move through the photosystem, we have already discussed in cyclic and non-cyclic photophosphorylation that when electrons move through photosystem, there occurs movement of hydrogen ions from, that is protons, from the stroma to the lumen of thylakoid. This is because the electron transport system is made up of cytochromes B and F, and they have a tendency of saying that we will transfer complete hydrogen atom as a whole, not as H plus ions and electrons individually. Therefore, electrons take the help of hydrogen ions to move through that electron transport system. So these hydrogen atoms are taken up from the stroma of chloroplast, and these hydrogen atoms will be sent to the lumen of thylakoid. And also the NADP reductase enzyme is present towards the side of stroma, which removes H plus ions from the stroma. Let me explain you it in detail. This is the diagram of chloroplast, the outer and inner membrane, and here we have granum, thylakoid, stroma lamella. So now we are discussing about the story inside thylakoid and stroma. Now what is happening is, whensoever electrons are passing, this is photosystem 2, this is photosystem 1, we all know this now. When electrons are passing from photosystem 2 moving to electron acceptor, then moving downhill through electron transport system, during this journey the electrons are moved through cytochromes B and F, and they have a tendency of using H plus ions and electrons coupled together. Therefore, now let's see what is happening. This is a core diagram of a thylakoid. This pink color membrane is actually a thylakoid membrane, and this thylakoid is present, let me draw it for you, is present inside the chloroplast. So if I draw it for you, so here would be a big chloroplast, which is a double membrane bound organelle. Now inside this chloroplast, this thylakoid is present, and when more than one thylakoid stack over each other, they form a stack which is called as grana. And right now we should focus on our thylakoid. Inside thylakoid we have lumen, and I told you that these photosystems are present on the thylakoid membrane. Now when this electron is passing from photosystem 2 to photosystem 1, as we can see in this diagram, electrons pass through photosystem 2 through electron acceptor to electron transport system to photosystem 1. There is electron transport system in between photosystem 2 and photosystem 1. And as I am stressing it so much that this electron transport system is made up of cytochrome B and F. Therefore, whensoever electrons pass through these cytochromes, they say, I'll not transfer electrons alone, I want hydrogen ions also. So thylakoids say, oh, better take this from the stroma. As we know that if it is a chloroplast, the surrounding medium is stroma and inside we have lumen. So from stroma, these hydrogen ions are taken up and sent towards the lumen when they pass through electron transport system. And finally they are sent to the reaction center of photosystem 1, as we can see here, would excite these electrons again to another electron acceptor, but now this electron acceptor will send it to NADP plus, which I am calling as a drug mafia, which will take these electrons and run away. So when these electrons are sent to NADP plus molecule, actually what is happening, you can easily see that towards stroma this reaction is taking place, and NADP would require H plus ion to fuse with it to form NADPH. Therefore, H plus ions are used from stroma and the concentration of H plus ion in stroma is decreasing. So two steps are increasing the concentration of H plus ions inside the lumen. Here you could see that water is splitting into H plus ions inside the lumen. There were three reasons for this proton gradient being developed. First is splitting of water occurs in the lumen, therefore H plus concentration increase in the lumen. Second thing is when the electrons move from photosystem 2 to photosystem 1 through electron transport system, these cytochromes require H plus ions for the transfer of electron. Therefore these are taken up from the stroma and sent it to the lumen of thylakoid, therefore increasing the concentration inside lumen. And the third reaction, which is reduction of NADP plus molecule, is actually taking place in the stroma, because the enzyme required for this reaction, which is NADP reductase enzyme, is present in the stroma of this double membrane bound organelle called as chloroplast. Therefore H plus ions in the stroma is decreasing. In stroma H plus is decreasing, in lumen H plus is increasing. Therefore there occurs a proton gradient between these two. And when H plus ions move from higher concentration, that is from the lumen of thylakoid to the stroma through this ATP synthase enzyme, it leads to the formation of ATP. So when these H plus ions will pass through this enzyme that is called as ATP synthase, there occurs phosphorylation of ADP into ATP. That completes the discussion. So here we need to stress that this ATP synthase enzyme has two parts. One is F0 and one is F1. Let us discuss about them in detail. We have another diagram from the internet which will make it crystal clear to you. So here we have this as a thylakoid, and inside we have lumen of thylakoid, and outside we have stroma of chloroplast. Now what is happening is water is splitting inside the lumen. You can see this reaction is taking place inside the lumen of thylakoid. Therefore H plus ion concentration is increasing in the lumen. This is the first reason for increasing H plus ion concentration inside the lumen. The second thing is when photosystem 2 excites its electron and wants to send it to photosystem 1, there occurs the transfer of electron through electron transport system, which is made up of cytochromes, particularly B and F. And these require H plus ions for transferring electron. Therefore, H plus ions are taken from the stroma of the chloroplast. Let me make a chloroplast again for you so that you could make a crystal clear image what is actually happening. So this is one single thylakoid present in a huge double membrane bound organelle which is called as chloroplast. So outside we have stroma, inside we have lumen. Now this is the second reason why H plus ions are increasing inside the lumen of thylakoid. And the third reaction is when photosystem 1 will excite the electron. You could see that in this image that photosystem 2 is sending its electron through electron transport system to photosystem 1. When photosystem 1 will excite its electron, it will be sent to an electron acceptor which will send its electron to NADP plus molecule. Now coming back to that story, NADP plus molecule is converted to NADPH towards the stroma. You see it is outside the membrane of thylakoid. Therefore they require H plus, which is taken up from the stroma. Therefore H plus in stroma is decreasing, H plus in lumen is increasing. So there occurs a gradient between lumen H plus concentration and stroma H plus concentration. And when these H plus ions move from higher concentration to lower concentration through an enzyme called as ATP synthase, there occurs the formation of ATP, and this has two parts. One is F0, which is the transmembrane part, and the other one is F1 part, which is bulging out part of this enzyme. Now let us talk about this enzyme ATP synthase. When this proton gradient is broken down, that is due to movement of protons, that means H plus ions, across the membrane to the stroma, across the membrane of lumen to the stroma through transmembrane channel of F0. I already mentioned it here that the transmembrane part of this enzyme is F0 and the bulging out part is the F1 part of this enzyme. So here we are seeing this gradient is broken down due to the movement of protons from the lumen to the stroma, and through this transmembrane part of this enzyme ATP synthase, that is called as F0 part of ATP synthase. Now this ATPase enzyme has two parts, F0 part, F1 part. F0 forms transmembrane channel, and through this occurs facilitated diffusion of protons, and the F0 part protrudes in the stroma and leads to the generation of ATP. Note that ATP and NADPH, which are produced in this reaction, is immediately used in the biosynthetic reaction which is taking place in the stroma. That is the dark reaction. So let us come back. Here we have seen that inside this stroma ATP is being formed because the F1 part of ATPase synthase enzyme is protruding inside the stroma. So this ATP is sent to the dark reaction which is taking place inside the stroma, and inside stroma we know that there are lots and lots of enzymes which will lead to the conversion of carbon dioxide into glucose molecule. And with this we have finished the chemiosmotic hypothesis discussion. Hope you have understood each and every detail. There are three important steps which lead to the formation of this gradient. First is splitting of water is occurring inside the lumen of thylakoid. Second is when electrons are transferred from photosystem 2 to photosystem 1 through electron transport system, which is made up of cytochromes B and F particularly, they require hydrogen ions also along with electrons. So these hydrogen ions are taken from the stroma and are sent inside the lumen of thylakoid, therefore increasing the concentration of H plus ions inside lumen. And the third and last thing is when photosystem 1 excites the electron and send it back to an electron acceptor which send it to NADP plus molecule, which this reaction is taking place inside the stroma within the presence of enzyme NADP reductase enzyme, which takes H plus from the stroma, therefore reducing H plus or say protons in stroma, and we have high H plus in lumen. So this high to low value, there occurs facilitated diffusion through transmembrane channel of ATPase synthase enzyme, that is F0 part of ATP synthase, and the F1 part is protruding out, which lead to the formation of ATP. This is the whole story behind photosynthesis, how they are making ATP. I hope it is clear to you. If you like this content, do check my other content also, and I'll upload the notes of this chapter on my Telegram channel. Thank you for watching this video till the end.